Hi, so I'd like you to imagine that you've just been infected with E. coli. The first thing that your immune system is going to do is it's going to want to recognize that E. coli is present. So it uses toll-like receptors, which are receptors that identify pathogen-associated molecular patterns on different pathogens. Toll-like receptor 9, or TLR9, is capable of recognizing the PAMP in E. coli that is uh, unmethylated CpG, which is on E. coli's DNA. So, what exactly is unmethylated CpG? So, on the DNA of E. coli, there are nucleotides sequences with cytosine, phosphate, and guanine. And in bacteria, the cytosine in this sequence is not methylated. This is unlike our own DNA, which when there's a cytosine, phosphate, and a guanine, or CpG, there is a methyl group on the cytosine, which is put there by DNA methyltransferase. So what does this all mean? So when E. coli is in your body, one of four cells that has TLR9, natural killer cells, B cells, eosinophils, or plazocytoid dendritic cells, are going to take in E. coli and are going to bind it to an endosome, which is where TLR actually is located. So the endosome is going to break down E. coli into its different parts, and importantly into its the DNA portion, because the DNA has unmethylated CpG. The unmethylated CpG is going to be able to actually bind to TLR9, and when it binds to TLR9, it's going to send a signal transduction pathway to the rest of the cell. This is going to cause the cell to send signals to the rest of the immune system that actually says that E. coli is in fact, or at least a pathogen, in this case E. coli, is actually present in the body and is going to host a defense against it.